Hello and welcome to Nikolai's genetics lessons, and the first question is which of the following molecules is or are produced by translation. So here's a double-stranded DNA, and here's a messenger RNA which grows continuously from 5' end to 3' end. And let's say here we have ribosome, large subunit, small subunit, and also tRNA here, which brings amino acids to growing polypeptide chain. So the process of making messenger RNA we call transcription, and the process of making polypeptide chain we call translation. So variant A, ribosomal proteins, would be made when polypeptide chain would make a three-dimensional form, and here's a ribosome. It has large subunit, small subunit, and it consists of about 50% of the volume of the RNA, scaffold RNA, and proteins. Not a single, but multiple proteins. So ribosomal proteins are result of the translation. So result of this process. tRNA or transfer RNA is a result of transcription, because tRNA is RNA and it's never going to be translated, it's only going to be transcribed. So we are looking for those molecules which are result of the translation. And variant C, the amino acid glycine. Glycine is going to be one structural subunit of the polypeptide, but glycine production or synthesis is different from the synthesis of the polypeptide or RNA, so it's not a result of the transcription or translation. So we also do not circle it. And RNA polymerase is going to be a protein that we can find somewhere here, which makes this RNA, and this is protein, and protein is a result of the translation. So we circle this variant of the answer. RNA polymerase is going to be another variant that meets our requirements. And the next, the digestive enzyme pepsin. Digestive enzyme pepsin is also protein, and all proteins are a result of the translation. So we also circle this variant. And second question, which of the following molecules is or are produced by transcription? Again, transcription is going to be a process of RNA production, not necessarily messenger RNA. As you see, it can be tRNA, it can be scaffold RNA, and even some RNA can be or function as enzyme. Will have enzymatic function or signal function. Also, some of the RNAs that never would be translated into polypeptide chain may have regulatory function. So let's check the list. Messenger RNA. So messenger RNA is a product of transcription. So we have to circle this variant A. And next, ribosomal proteins. Ribosomal proteins would be result of the protein folding of the polypeptide chain folding, and polypeptide chain have to be produced first by the process of translation of the messenger RNA by ribosome. So this is a result of the translation and not transcription. So we do not circle this variant. Ribosomal RNA, on the contrary, take a look. So this is large ribosomal subunit. 50% of the volume is made by RNA, and another 50% by different proteins. So I'm going to use different color, so you know that there's many different proteins that make large and small ribosomal subunits, and ribosomal proteins would be result of the translation and ribosomal RNA would be result of the transcription. So we are looking for transcription. So ribosomal proteins we do not circle, but ribosomal RNA we circle. This is result of the transcription. And the last variant, transfer RNA, as you know, is never translated into the protein. It functions as RNA, bringing individual amino acids to growing chain of the polypeptide, to ribosome, to messenger RNA. So transfer RNA is a result of transcription. So we circle. This is also correct statement. And this is all for today. Subscribe and see you in the next video. Goodbye.